Okay, everyone, welcome to part one, framing your research. We're going to start by just sort of laying out the general framework on how you might begin to take your entire thesis research and turn it into a 3-minute thesis pitch. So the challenge, of course, is communicating your research to the public. And it is a challenge because when you are taught to become a researcher, when you're doing a PhD, you're not only taught how to, conduct certain experiments and stuff, you are taught all of the nuance of science. You are taught about how your field understands your topic. You are taught how to explain and write about your research in papers. And there's a whole process of becoming a researcher. And what you're actually being asked to do with the 3-minute thesis is sort of reverse that and take everything you've learned and these scientific skills and tools and reverse it into a neatly packaged story that anyone can understand. The best approach, I think, is to find the right framing, and then you're going to translate it. So just like you could take any section of text and translate it into the right sort of written format that you might use for a research paper. You can do the exact same to go backwards, and that's what we're going to talk about. So in order to choose what the right framing is, you need to ask yourself, what makes your research interesting to non-experts? What about it would people become excited about? Without their attention, you can achieve nothing. So I've learned this through my social medias. Of course, if people are not engaged and interested in your research, then you won't be able to tell them anything. You won't be able to teach them anything. And of course, I'm sure you've experienced this as well in your classes that you've taken throughout undergrad and your PhD now, and maybe even throughout high school. When you're in your least favorite class, you find it very difficult to learn because it's very difficult to pay attention. Whereas your most interesting, your favorite classes, the knowledge just sticks naturally. So to find the right framing and present it to them in a way that they will be interested and excited about it is going to help them it's going to help the information stick. So to find that, you might ask yourself, what is the most compelling or impressive finding? Understanding this will be the basis for your entire presentation. The most interesting finding that you have is your core for your presentation. Now, if you are not at the stages of your project where you have interesting findings, you can still compete in the 3-minute thesis with just sort of either preliminary data or no data at all, in which case you would be talking about a proposal that you have. Maybe you say, we've found this one piece of preliminary data, and we believe that it's going to help us understand this, and this is how we're going to do it. In that case, it wouldn't be your most impressive finding. It would be like your most interesting goal. What's the most captivating goal of the entire research project? So think about these. There'll be an activity at the end of this for you to ask yourself this and take some time for this. You'd also have to ask yourself, in general, what parts of your research would the public naturally understand and what wouldn't they? So whatever falls into the latter category must be translated, and we're going to talk about that. So finally, I'm going to finish with a comment from myself. To successfully communicate your research to the lay public, you must be both interesting and interpretable. This, of course, the 3-minute thesis focuses on the interpretable part, but I think that you must also consider the interesting part. Okay, so before we move on, I just want to include this note for those in the humanities and social sciences, but this applies to everyone, so please don't skip this section if you're not in the humanities and social sciences. So I talk a lot about this idea of translating your research. And as a person coming from a biology background, of course, there's a fair amount of translation required for presenting my research. Not everyone knows about synapses and how brain cells communicate and all this stuff. And there's a general sort of belief that biology and chemistry and these really like sort of molecular sciences require a ton of translation and maybe require more than other fields. But I want to say the level of translation does differ by field and by presentation, but it's not only required for biology and chemistry. So you may think, let's say if you're in the social sciences, since it doesn't require like this deep knowledge of molecular function, that it will be more generally interpretable to the lay public, but that may not necessarily be true. Every one of us, our research has some level of overlap with general knowledge that the public holds. that general knowledge depends on what really everyone is taught in high school and college and whatever you can expect the average person to have as far as education. So a good example of this is mitochondria is the powerhouse of the cell. Everyone knows that. But if you actually start talking about mitochondria and you get into, how they work on like a molecular basis, no one knows that. So you just have to ask yourself, like, what parts of my research overlap with that knowledge? And even if it does overlap, I would still recommend giving just like a quick refresher and including those points. So if there are parts of your research that seem obvious, and when I say obvious, I mean like that seem to fall into that category of everyone knows it, you have to ask yourself, why is this obvious? And really, really, truly think about it. Maybe this is a good time to start asking a friend or a parent or grandparent, someone who is not an expert in your field, if they know it and if they understand it. Because a lot of people fall prey to this trap of being really familiar with the scientific literature on a topic, on their topic, their research, your research topic. And you think that, this is just so foundationally built into my understanding of the world that everyone else must know this too. But that's, again, not the case a lot of the time. So, and there's a good example of this where I was doing this workshop for another university and a student asked me, hey, my research is just, it's so clear, everyone already knows it. What do I do? Do I still translate it? And we started talking about it. And it turned out that the students' research was, what they had found was replicating a previously published study. And she had gotten into this perspective that because it's already published, like everyone knows it. But as we're talking about it, I was very clear. I'm like, I don't know this. I'd never heard about this before. This is news to me. And it's really great that you're replicating another study, that shows that whatever that finding is pretty strong and solid. But just because it's been published before doesn't mean that the general public knows it. So ask yourself, why do I think that this is obvious? And would my grandma think it's obvious too? And literally, you can ask your grandma if she thinks it's obvious. So another point is that not everyone is as inclined to these topics as you are. So of course, you have chosen to do a PhD in this topic, probably because you like it. And if you like it, it probably means you're good at it. Just like I was saying earlier, that a lot of people sit in classes and have, it's troubling to pay attention to classes you don't find interesting, and it's easy to pay attention to classes you find more fascinating. Some topics just resonate better with us naturally. And so if something seems really clear and obvious to you, it may not be clear and obvious to another person. It might just be because you really have a natural knack for understanding this subject, which again is why you've chosen to do a PhD in it. Maybe there's some deeper psychological stuff going on there. So ask yourself, is this just my way of thinking, or is this everyone's way of thinking? So with that said, you understand it naturally, but be aware of the viewer's level of understanding. And again, this is a great place where you can solicit advice. This is not a good place to talk to other students in your PhD program who have the same training. This is a great opportunity to go to, anyone, go to your gym and ask someone who you work out with, or, someone you live nearby, your neighbor or friend, anyone. What do you know about this topic? And actually, what I really would recommend is just explain it to them. And after this presentation, if you have a 3-minute pitch, ask them, hey, can I borrow 3 of your minutes of your life? And let me sit down and read this to you, and ask them to point out every part where they get lost, because that's what's going to take your presentation and make it, worthy of winning the 3-minute thesis, because you don't want to lose the viewer at any point. So when you do choose to translate, you have to decide, maybe decide isn't the right word, but you have to balance over and undersimplifying. So if you undersimplify, of course, you run the risk of them not understanding it. If you oversimplify, you run the risk of potentially insulting someone. And I think it's also good to think about each topic that requires translation as like a scale of 1 to 10, how much translation is required. This was my paper title. So this is one of the papers in my PhD thesis. And I'm not even going to read it. It's gibberish, right? So if you're not a neuroscientist, synaptic behavioral deficits and 16p11.2 duplication, like what is going on here? So worse than that, here are some of the data from my paper. And I mean, if you're not already nervous just looking at this, Clearly, there's a lot going on here. And without knowing anything about this, this is absolutely uninterpretable. There's no way you can understand what is going on here. So I had to start at the very beginning, and this is what I'm going to ask you to do. Break it all down. So you need to take what your research is, all of this, and break it down into concepts, not graphs, not data. So in my case, I was studying a genetic mouse model of autism spectrum disorder. Okay. There were changes in the behavior of the mice. There were changes in social behavior, which makes sense. So autism spectrum disorder is a condition that affects social behavior in human beings, of course. So the mice also showed these social changes. There was also changes in cell-to-cell communication in the brain. And by the way, don't worry, you're not going to be tested on this. I'm just showing you how I laid this out. This is going to turn into a lesson. There were tons of supporting biochemistry and sequencing data and all this stuff. We identified a key molecule that was acting a bit strange in the brains of these mice, and we found the same change in human autism spectrum disorder brain tissue. And finally, creating more of that protein, the molecule made the mice more social and improved cell-to-cell communication. So I want to ask you, which of these points would you consider is the core? Remember, we talked about identifying the most critical, interesting finding. And that's a challenge in itself, identifying what the public would find interesting. But I hope that here it will be fairly obvious. To me, it's this right here, that essentially we were able to elevate the sociability of a mouse model of autism spectrum disorder and improve this change in the way the brain was functioning. Why that's the most interesting, it's very hard to define. I think that generally the most, in the media, the most catchy headlines are like new treatment for cancer discovered. And this sort of conveys that same sense. But to me, this was clearly the hook for my presentation. But in order to get to this, I had to first explain a lot of foundational knowledge. So I had to explain this. I can't talk about how it improved the social behavior in the mice without first explaining that there were baseline changes in social behavior and in cell-to-cell communication. So these 3 points together seemed like, my foundation. And this is why it's important to break your research down, because you go from what I showed on the last slide, a mess of data and figures and graphs, to 3 key points. So in a minute, you're going to be asked to do this yourself, but I just want to finish going through this. So going to point number 1, I didn't feel that it was necessary to talk about the fact that it was a specific genetic mouse model. 16p11.2 duplication is the genetic change, and I just felt that it was going to be overwhelming to include that information. I also think that once you start introducing acronyms, 16p11.2 duplication, people get confused and start to sort of try to remember what that piece of information was, and they're using some of their mental energy on that, and they kind of miss the rest of the presentation. So I would just recommend leaving out those things if you can. And simply put, in this case, I just said I was studying a genetic mouse model, or a mouse with a gene mutation that is associated with autism in humans. They don't have to know what the gene is. Of course, number 4, I do not want to talk about this supporting biochemistry and sequencing, anything unnecessary, anything that doesn't contribute directly to my main story doesn't need to be talked about. And lastly, number 5, this had to be sort of included. So I don't bold this, I don't gray this, but this ended up being a key part of the presentation as sort of like a segue to how I explained it all. So activity 1, break it all down. I want you to take some time, if you are willing, to ask yourself, what is your research all about? Just like I did there, where I took my research and I broke it into just 6 bullet points, the key outline. Please try to do that for yourself and define the broad topic. Ask yourself, why are you doing it? Ask yourself as many questions as you can to help produce just a short list of like, here's the start, here's the rationale, these are the main findings. And then try to identify like, what is the key topic within that. And then ask yourself what framing could work for you. Like we've talked about, what makes your research interesting? What are the most compelling or impressive findings? And then of course, what would the general public understand and what would they not? So I wish you luck. Please spend a few minutes just writing this down. Again, you don't have to. And if you're going to do this activity, it will help you throughout the rest of the activities. So I encourage you to do them all, because again, you will end up with a pretty much written 3-minute thesis at the end of it. So good luck, and I'll see you in part 2.